So this panel is about how small and large companies are tackling creating efficiency in trials so that they can advance despite the current difficult financial situation for biotech. And so the idea was to have both representation from expertise from a small biotech, but also somebody who sees things from the other side, an investor. Because more and more in this, especially in the last year, given my changes in what I've been doing, I've been quite interested in how things are seen from the other side. When I left academia, they were telling me, "Oh, you're going to the dark side. You're going to industry, and now it's going to be all corrupt." And actually, I met a lot of people who wanted to develop drugs and help patients. And then in biotech, sometimes they say, "Oh, the investors, the sharks." And now that I've been talking with investors more and more, I see investors have a dilemma. Where do they put the money that somebody is giving them to invest? Somebody gives money to the investor has to make it grow. It's our pensions in our retirement funds. So if we look at it if we put ourselves in the other people's shoes, then we realize everyone has difficulties in what they're doing, and everyone is trying to do the best thing possible. So the idea was, okay, there's a financial limitation at the moment for biotechs and restricted amount of funds. And so we can, of course, chat about this. There's no dogma, but let's throw out some ideas on how to make these trials efficient and how does it look from the other side, from the investor point of view. So before we start this chat, it's a fireless conversation, fireless chat here. I think it's helpful if we briefly introduce each other, starting from Lisa.
Yes, thank you. Thank you for the invitation. It's such a great opportunity to talk with such a diversity. I'm Lisa Mahnke. I am the Chief Medical Officer for Nerviano Medical Sciences based in Milan. We have a very small office here where I'm the CEO and the president. And so we're a European company. I've been in big pharma for almost 20 years. I've done about a third of my time in very large pharma, about a third of my time in mid-sized pharma, and about a third of my time in small pharma. So I've seen it from all the angles, and I've seen some surprises moving from big pharma to small pharma that I'll comment on.
Great. Hi, my name is Chris Bardon. I'm a physician by training, but now I work as an investor. I'm the co-head of MPM Capital, which does private and public investing in biotech. Nice to meet everybody.
Well, let me add, they're very modest, of course, about themselves. But Lisa, the companies, can you name the companies where you were before?
Yeah, let me give you the long list or the short list. So I've worked with Bristol Myers Squibb. I've worked for Merck MSD. And then I made the move to Vertex, which brought me to Boston, which I'm delighted about. I'm so happy to be here in Boston. And then I worked for Merck KGaA, the other Merck. I worked for Syros Pharmaceuticals. I worked for Bicycle Therapeutics. And I worked for Servier down in the Seaport, also in France. And now I'm with Nerviano. So I feel quite lucky because I've got a very, very good European view. I have a lot of hands-on experience in Europe from the European mindset, European regulators, European operations, European staff. And I feel like that helps me a lot because I heard in the prior discussion, we always have to kind of bridge across the Atlantic for sure. And so I'm kind of in the opposite boat. I'm in Europe mainly and then trying to, bridge to the US.
And you're a physician and you used to work in kind of pharmacology, right? And
yes.
then you are the person who led.
Yes, you're so
bring
kind.
It to market.
He's so kind. So I'm a physician scientist, MD, PhD, and I got in at a time when IO was just starting. So I started with Bristol Myers. I'm an HIV doctor by training, and I was hired to bring that immune component. The host response to PD-1 was discovered in HIV and Hep C before it came to tumors. And that's why Bristol Myers hired me years ago. That's when IPI was making the move from phase 1 to phase 2, phase 3. And they had a 4-1BB that they wanted me to work on. And so I was lucky to get into IO at those early days when everything was just so novel and so cool. And then I've stayed with oncology all those years. I've been on and off. I've done small molecules and large molecules. And you're so kind to mention, I have tried to bridge myself between clinical leadership and clin pharm leadership. And I have served as the global head of clin pharm for Vertex and Merck KGaA. And I've done a lot of translational leadership in addition to my other roles that I've done over the years as a clinician doing regular clinical leadership. I was the main clinical leader for the first approval of Bavencio for Merck KGaA, and it was such a special time. I did all of the lead negotiations with the health authorities, and it was a very special and amazing time. So I was lucky to be in that role. But what I try to do is I try to use kind of the two sides of me, the clin pharm side of me and the clinician side of me, because it speeds me up. I can speed myself up sometimes. If I encounter or I see a problem coming, like, for example, we're talking a lot about Project Optimus these days. I hear a lot of what I consider to be myths around that. We have to pause for Project Optimus. We have to do all these randomizations for Project Optimus. It's going to take a lot of time. There's going to be a lot of gaps, a lot of pausing. But in my mind, that's really not true. And that just comes from me being a clinical pharmacologist as well, where I know how to make those arguments with the FDA on PK/PD. I've done it so many times. And I just know what the FDA is thinking. I've sat with the Office of Clin Pharm at the FDA many times. I've been part of the IQ group. I've been down there a bunch of times, a bunch of things like that. And so I feel like I can use my experience, and I try to use this cross-functional nature that I have to just help myself be more efficient and then guide my teams to be more efficient and to really understand what the FDA and the EMA are thinking. I hear a lot of myths around that. But that would be one way to try to make us more efficient, to move ourselves forward, is by being smart.
And I wanted to mention the Bavencio experience in registered, because Bavencio was registered first in Merkel cell carcinoma. That was the first application, which was unusual for a PD-1, but then cemiplimab did the same, Libtayo, right, in CSCC. So use a small, rarer indication to accelerate finding data, which is very much on topic what we're discussing today. And Christiana runs one of the most well-known and renowned investment funds for small biotechs, especially here in Boston. And she sees lots of companies and what they're doing. And so that's why we thought it was precious to have her here, because she can tell us, without naming names, but how are small companies accelerating data or getting data with less, maybe cost patients faster in a more nimble way. And for myself, I'm an oncologist. I was an investigator in clinical trials from 2001 to 2005, phase 1, 2, and 3. Then I came here for a PhD in genetics. I've been 10 years in small biotechs, and then 7 at large pharma Sanofi. Overall, I directly worked on about 30 clinical drugs, phase 1, 2, and last 3, of which 26 I wrote the protocol, put myself in the clinic, plus some I was responsible for preclinical, so 3 years before the clinic to the clinic. And then you may have read how Sanofi is reducing investment in oncology, so this summer I decided to change, and I'm now Chief Medical Officer at a small biotech here in Boston, ModeX Therapeutics. The point is, well, maybe we can first hear from the clinical development side, and well, you represent not only clinical development, maybe I represent clinical development, you represent development, and also the cost and the business side of a small company. And let's see if Christiana agrees with with our approaches, or actually, better, she can add more suggestions on.
I think that's great, because I've heard you speak before, Christiana, and some of the things you've said have really rung true for me, and I've tried to take those things back and emphasize. Let me give you just a couple of points. Two of the biggest surprises that I see as I move from big pharma to little pharma is in oncology. I'm really speaking strictly on oncology, although I have worked in other therapeutic areas, and we could talk about other therapeutic areas if you want. I've been around several NDAs and BLAs and been primary on a couple of those, including Bavencio. And I've been around a lot of end of phase 2 packages and lots of INDs, many INDs. So that's my perspective. But when it comes to oncology, one of the biggest myths that I see, which surprises me, is not wanting to embrace the seamless pathway. Okay, seamless pathway is designed for efficiency. And we're here to talk about efficiency, right? So for me, it's super logical to want to leverage the seamless pathway in oncology, okay? No downsides really to launching, early on accelerated approval and following on with your confirmatory study in an earlier line. To me, that's fairly obvious because it's fast, it's efficient. And I heard, I think it was Michelle, you were saying something about be aware of standard of care changing. Standard of care changing is a killer in oncology, and it happens every 6 months. Something changes in oncology in any tumor that you're working on. So what we tell our people, we teach, I have a series of slides, we call it Oncology Forward. And it's that same concept that I heard you mention is look forward, anticipate, because if you do a single arm development and you can emerge like we did with Bavencio on 88 patients, very rapidly, very quickly, and you're following in with your life cycle and your confirmatory study and you're expanding your label in other ways, that is great because that's one way to beat the changing nature of the landscape. If the standard of care changes, now you're going to have review issues. You might not have relevant data. And, let me give you an example. There's a drug that I won't name, but the median PFS in the small early study, single arm, was estimated at 30 months. But by the time they got to the large randomized study, I believe it was 800 patients, that median PFS was only 5 months. Okay. Now there's two problems with that. One is a lot of things change year by year as you go during development, especially in oncology. Patients can access so many other things, just their basic standards of care, basic palliative care, basic supportive care, or their chance to maybe access something emerging off-label or something in the US. So many things that change. And the FDA always says this, and the EMA as well. You cannot interpret median PFS on single-arm data. This is one of my hugest pet peeves that I don't like when I see this, and I see big companies making this mistake. I see small companies. I hear people telling me, this is what we want to hear. We want to hear what that median PFS is. But you have to be stringent on how you use data, okay? Just because we're in the clinic doesn't mean we stop being scientific about how we collect data and how we analyze and interpret data. You have to interpret. You can't interpret single-arm median PFS or OS, and the FDA tells us that, and we know that. It's not scientifically valid, really, to do that. You can make a big mistake, and I can give you many examples where companies have failed in phase 3 by doing that. Okay. So that's another reason why, and the standard of care could change too, which would be another big reason why you could fail. Okay. So this is why in oncology, to be efficient, we want to still use the single arm pathway. And I hear a lot of myths around that. Oh, the FDA doesn't want that anymore. For checkpoint inhibitors, they said they kind of didn't want that anymore because there are too many, right? But for everything else, every 6 and 8 months, we see another one emerge. And it's always emerges on the same pattern. It's always let's say 60, 80, 100 patient with a good lower bound relative to some historical standard that's very, very meaningful and very compelling. Okay, not just a simple, looks better. It can't look better. It's got to be really compellingly better. And then the FDA reviews that package like they always do. They're very consistent on how they review those packages. And you still see these single arm approvals emerging. So one of my points is...
Let me put a dent into this, which is since last year, I think, FDA started asking that you only get an accelerated approval if you complete enrollment of your phase 3 study and you have some initial data from the first 3 study. That was not the case before. So getting an accelerated approval today is absolutely possible. It becomes more challenging for a small biotech who cannot afford to launch a phase 3 study. So you do need a partner with a large pharma or larger funds in order to achieve accelerated approval because you need to have already almost completed enrollment of a phase 3.
That's true. That's a very good point. For Bavencio, we were the first company to negotiate with the FDA to get a single arm confirmatory study. That had never happened before. But as a small company, you're right. You have to be super careful about that design of that confirmatory study. It can't be so huge that you can't afford it or you can't enroll it. That's a really good point. And yes, the FDA is more stringent now about, whereas before they would say 50% enrollment at the time you file your NDA or your BLA. Now they're saying it's got to be closer to 100%. But, especially if you're working under breakthrough, for example, if you have a highly novel drug and you work under breakthrough, the FDA, and I've worked under breakthrough at least 6 times now, you can really talk back and forth with the FDA. And I think if the unmet medical need is very high and your product is very compelling and is really serving that unmet medical need, I heard someone mention that earlier as well, I think the FDA will be willing to work with you on that, and to have a very good, relevant, confirmatory study designed well. There are some ways to do add-on to standard of care versus no add-on where the sample size is really not that large, like a maintenance design, a switch maintenance design, for example. So there's some ways you can still approach that and have it be manageable. But I hear what you're saying. You might need to have a partner.
So we started all the way from the end, which is the approval. Let's go a little bit back in time. Let's do in order, right? Because I think we should touch on what preclinical data are actually needed to get into the clinic. That's maybe different from what preclinical data an investor may want to see to actually fund a company. And then how do we get to, when I joined Sanofi at the beginning, when I took over I launched the early development team, and there was 2 projects ongoing. And for a phase 1, they had a plan for 500 patients in the phase 1 study, all comers. Let's test all indications, multiple doses, and then depending on the signal, we see where we go. It was not today's time, but we changed entirely the approach. So how are you planning early data reads and early signals from an phase 1A dose escalation trial? And then what's the investor view? What type of clinical early data would you like to see to fund a company? So let's do maybe the same step. Maybe we'll do one at a time. First, we do the preclinical, and we ask also the investor. Then we do the clinical, and we ask also the investor. You wanted to add something?
And
a question? Oh, okay.
The translatability of the preclinical data. If you're talking about a checkpoint inhibitor or IO or immune modulator, it's a lot different than a targeted or a small molecule. So let's talk about the target and small molecule. We need to see a signal. We need to see it in a meaningful situation. Some animal data, some type of animal model, at least for a first in human. But we can't extrapolate too much on that. So what I do is when I look at the packages that come to me, I'm looking for some cell line data that might help me and guide me a little bit. I'm looking for at least one animal model that seems to be relevant. I'm looking for not just tumor stasis, but regression. And I'm looking to make sure that it's really causing the effects on the tumor that I know I need. I can't just hope. I can't just say, I hope in the mouse that I'm getting tumor stasis, but maybe in the human, I'll get something better. No, I want to see something a little bit more compelling. But I have to be careful because those primary models come from different types of cell line or patient grafts that are not always applicable to what I'm going to be doing in the clinic. When I get in the clinic, I'm dealing with patients who have failed multiple therapies and who have other cofactors or other types of genetic factors that I didn't see.
So, but then especially if we look at immuno-oncology agents, where not only you need to kill the tumor, but you need to activate the immune system. And there are, of course, syngeneic mouse models, and it's all very artificial. What is the minimum? Because, if there's no activity at all in a mouse, you don't bring the drug forward at all, right? If there is some activity, it's more to say, okay, we don't kill it, we keep it. But it's not that you're gonna, I've heard people trying to do 5, 6 different models so that they can show beautiful preclinical. It doesn't really matter in the clinic because you're not going to chase your indication based on that because of the differences between even a good mouse model and patients, especially with IO.
Especially for IO. So IO is not nearly as predictive moving from the preclinical to the clinical. And we just have to be aware of that, right? And I think actually we have an advantage going for us right now because the models are not predictive. You can't use a nude model because then you don't have the immune system. And even these transplanted models, it's all artificial. Plus, you've got the MHC restrictions.
And some receptor not even cross-binding, right? So the mouse receptor will be different than the human one. So, basically,
it's never.
You agree
predicted.
that do the minimum possible just to not kill the molecule. Then try to go as fast as possible to the clinic. Now that was true 3 years ago, and companies were getting funded with that data 3 years ago. Today, if I come to you with a good preclinical molecule, the minimum amount of data, because I don't want to waste time or money, and I want to go to the clinic, what do you say?
So the first thing that we didn't really talk about is the biological rationale. So, we kind of glossed over that, but that's my number 1, 2, and 3 important thing. Because especially in oncology, preclinical models are not predictive of anything, right? I mean, how many failed MC38 positive drugs have we seen? So the biological rationale, and what I'd like to see there is genetic validation in humans, right? So if you, for example, even if you're looking at autoimmune, there are IL-1 variants in pericarditis and whatever, various diseases, which give you some hint of where you might see efficacy. So I like to see target validation based upon human genetics. That's one thing I really like.
So more than the in vivo model, you focus on the basic science rationale and a clinical data validation.
Let's start there. Now, different disease areas have different in vivo animal models, some of which are more relevant or not. I would say in oncology, they're essentially not very relevant. I mean, of course, if you don't show tumoricidal activity, then you got nothing, but showing tumoricidal activity is not necessarily predictive of future success. So different disease areas, other disease areas are more predictive, so it really just depends. Antibiotics are very predictive in a preclinical model. I'd say the next thing we want to make sure is we have clean tox, right? So what we do understand is that NHP tox is roughly predictive of human tox. The problem is NHPs are harder and harder to get, very, very expensive. And now there's a little bit, sometimes there's even, let's say, I think we had a policy where for a while we couldn't get access to certain species, and then we had to shift species, and there was a shortage. So again, NHPs have become, so there are other animal models I think people are using, depending again on the disease area, that have become very important. I think also the other thing is important is really understanding drug properties, right? So at the end of the day, a lot of people go into the clinic with just a dirty, bad drug, right? So does this have clean absorption, linear absorption, dose proportional exposure, really the basics. That's not so much of an issue in a protein therapeutic, but certainly for a small molecule it is. On the early clinical side, Elisa was mostly talking about late clinical. I would say, our companies have to get to proof of concept before they run out of money. That's the goal, right? I need to show I have a drug that works before I drop dead. So how can you do that? So a few questions. One, I think a biomarker is an absolute must nowadays, right? And usually that ties to the molecular rationale of your drug and hopefully predicts a population in which your drug actually works. So, everyone has a companion diagnostics. You can even say maybe Prometheus got acquired because they had a companion diagnostic in autoimmune, which we've just started to do, right? So that's an incredibly important. I think understanding biomarker positive things. Also translational biology, right? So based upon the biology of the target and what you expect to inhibit or change or augment, we need to have clear markers of that pharmacodynamically in patients to show that your drug is doing something, right? Because even if you do get positive clinical data in a single-arm setting, I don't really know if that's truth or random. That's why single-arm trials don't work. You could have just gotten a random bunch of patients that happen to live longer than they would have. And so again, the ability to correlate single-arm data with the biology, right? So if it's a vaccine, you say, hey, these people got the highest immunogenicity, and look, they also live the longest, right? That is the kind of thing you need to show from a translational perspective. On a checkpoint inhibitor, you can say, oh, well, there's, Tregs are going up or down, biology is doing this or that. And again, also that might require, for example, tumor biopsies, right? We don't get enough tumor biopsies. We don't get enough tissue biopsies to see whether we have proper drug exposure in the target tissue or whether the biology is changing in that tissue. So I would also pay a lot of attention to translational work, which I think we minimize. And then the final thing is, with regard to patients, you gotta be like a hawk on your clinical trial, right? So why do single-arm PD-1 failure trials not predict the future? It's because all those trials let in a bunch of patients who aren't really PD-1 failures, right? They're, either they progress, but the tumor went from 0.5 to 0.75, which isn't really much of a progression. Or, for example, they actually dropped off their PD-1 because of tox and not because they actually failed. So we've gone under confidentiality to review supposedly PD-1 failures, which actually aren't PD-1 failures, which means when I review your data, it's really not even relevant at all because they're not PD-1 failures. So therefore, the single-arm PD-1 combo data is worth nothing, right? So the whole point is if you're going to spend money for a clinical trial, make sure you are enrolling the right patients. Review every single patient that comes into the trial. Make sure they're the ones you want, right? So if you want PD-1 failures, I want to see progression on the PD-1 of the tumor looking like this, and then I want to see the tumor going down after you initiate treatment.
Have you ever seen that?
Not too often, which is why we don't invest in checkpoints too often. But the whole point is you need to be like a hawk on your patients, right? You need to review, for example, if there's a biomarker strategy, that they're really, really positive. And then as well, the only people that tend to get through on these single-arm strategies are people that have biomarker-identified populations.
So you went a little bit ahead beyond the preclinical, but that's okay. You gave us a window into the next part. But actually, I was thinking between the preclinical and a smart clinical planning and execution, there is the launch of the clinical trial. So maybe let me start from you this time. The amount of data to be submitted to different health authorities, the starting dose may change between country and country that the country would agree. The design and the commitment to phase 1B may differ from country to country, especially with Project Optimus now still. I've developed and I've seen other small companies skipping the US for the first in human. Many have flocked Australia, but even Europe or the UK. And we can go into the details, but first question for you is, some small companies especially are afraid of going ex-US for the phase 1 study because they're afraid that if they don't have a big US site, the investor will not take as seriously the data coming from Australia or Europe. What do you say to that? Is that right? Do you want US sites in trials, or as long as it's Western world sites, is that fine?
I don't think that that's a requirement at all. In fact, all of our gene editing companies go to New Zealand. For whatever reason, it's easier to get into the clinic in New Zealand. And so for gene editing trials, and again, as long as they practice Western style standards of medicine, I would say even China data, which people, obviously sometimes poo-poo, although now we know that data can sometimes be very, very predictive, it really just depends on the clinical trial site, right? It turns out that there are a certain number of sites in China that have high-quality investigators and turn out high-quality data. So it really just depends. I don't think it's mandatory to have a U.S. site.
Thank you. And so, Lisa, as a background, and I want to know where you do the trials or where you have done the trials, but it's very, very important to stay abreast of the latest regulatory situation. Project Optimus, yes, you can answer the FDA, but in the protocol design, they may tell you do not open an expansion cohort until you demonstrated that you're optimized the dose, which means double the number of patients before you can actually go and much more time before you can go to an expansion cohort. And for an IO drug, means a lot of risk because they ask you to choose the indication to optimize without first checking which indication is the right one. So that's one reason. Then US sites, and then for biologics, they ask you to start from a mAbs dose, a microdose, which other countries don't. So a lot of companies have gone to Australia to the point where Australia is overflooded with studies now. And I'm not sure that the timelines are always as good depending on what type of population you need. In the meantime, Europe tried to make it more difficult by harmonizing the process for Europe. It's good for phase 3, but for phase 1, now the minimum timeline is 90 days, if not longer. And if you submit to at least 2 or 3 countries, all Europe starts discussing, hey, no, I like it this way, no, it should be this way. And any country can stop and delay your process unless you go to a single country in Europe. Eastern Europe is still a little bit different. And now we have the UK. Oh, Brexit. Let's take advantage of Brexit. Who cares about this European regulation? And by the way, last time I submitted to the UK, they told me, no, we're not going to start your study here because the starting dose is too low. Too low. Yes, we don't want you to start until you get a pharmacologically active dose because we don't want our UK patients to take the equivalent of placebo for a cancer drug. So if you go just to UK at the beginning, you may skip 3, 4, 5 dose levels because they want you to start close to the pharmacological. So with all this in mind...
These are all good points. These are all good points. And I've had these discussions with many companies and many people. My perspective is a little different. I've had success with the FDA where I have wanted to have it. Okay, now with biologics, they'll push you to a really ultimate kind of MABEL definition, especially if it's a CD3 bite for sure, 100%.
And especially if the animal model cannot be really used because the epitope is different than the human.
So biologics with something sensitive on a T cell with hyperactivation like a CD3 or the old T Gennaro issue with the CD28, those are for sure. And maybe some other products like specialized products. But everything else, including biologics, I have had success getting the starting dose that I want. And yes, I had the same experience with MHRA where I joined a company. I told them they had their starting dose too low. I went to MHRA and US FDA both after already the pre-IND packages had been there. And I said, no, we're going to raise the starting dose because the starting dose is a logical calculation that you do based on the EMA 2017 guidance and first principles. As long as you use the first principles, you make the arguments and you extrapolate back to how you make your arguments relative to the preclinical tox. I thought that was a great point. As long as you make your arguments the way the FDA and the EMA want to hear it and the MHRA, they're going to back you up. And I had the same experience. I said, we're going to raise the dose. They said, fine, that's no problem. But I used the first principles of how we're supposed to do that. So that's one thing. So I am not afraid. Now, I'm not working on a CD3 bite, and I'm not working on something exotic like a gene therapy or cell therapy. So for those areas, maybe not. But I have had success, even for a biologic, even a kind of a complex biologic, like a fusion with a PD-1 with something else. I've done twice. I've been able to get the exact starting dose I wanted. So I'm not afraid to do the FDA. And I like the FDA first in human because I do think it sets a tone. I know that 30-day window. I know exactly how to use it, and I know exactly what to do. I know exactly if FDA is going to come in with some kind of clinical hold issue, I know they're going to do it during the 30 days. And I also know how to make arguments to make sure that I'm really buffering against that anyway. And then I'm ready to go with my dosing very shortly after that 30-day window. There's a lot that you can do in parallel during that time. This is where I've seen mythology come in. Mythology, when I moved from large companies to small companies, I heard a lot of myths about first in human, because you asked me about how can we go faster there. When I see a company that has filed their IND, but then announces a year later they've dosed, I know right away there's something wrong. Obviously, there's something wrong. Now, maybe they just had bad luck with their drug supply or something, maybe. Or maybe they just weren't on top of their CRO or their enrollments or something, or their sites to get things really moving, because there's a lot that you can do in parallel. And I hear these myths. Oh, you can't submit to the central IRB until you have this. You can't do this until you have that. Not true. Not true. I'm here to tell you. I learned from Bristol Myers and Merck MSD, fundamental principles of how to just get those things fast. And we even did a deep, deep task force at Merck KGA on, how to just keep all of those early processes moving. So my standard is we file, we anticipate the 30 days. A few weeks later, we are definitely bringing patients in for screening for sure, and even potentially dosing. Now, I liked your point about being stringent, and that was going to be my next point is...
But before the clinical execution, so US, you can manage. What about alternative countries? Do you prefer a Eastern European country, a Western European country, Australia, UK? Who's your second?
Well, the CTR has changed that because now the major Western European countries, so obviously, we think about Germany, Italy, France, okay, I'm still comfortable working with all those countries because I've done it for a long time now and I've worked for European companies, and the CTR has changed it though. You're right. You're exactly right. It's pushing you back a few months by default for sure. So what we're doing now is we're just trying to stage how we're doing things. We're doing some things here in the US at a certain time. We're waiting for Europe to come online. And then we're going to go back and use Europe for other things. And we're just trying to play off of the two regions.
Have you tried
the speed?
Have you tried submitting first to a single European country so that you don't get into the sense of the CTR and then amending to add more European countries?
We're doing that now, but in a kind of a back-ended way. I'm working with Switzerland, and that they're on a different time frame, not tied to the CTR. And then we're using the CTR where the other countries are tied in. But that all changed. That changed within the last 8 months or so. So in a sense, yes, we are. And I think I would do that again.
Switzerland is part of the CTR?
We're using them outside of the CTR.
Yeah. So no, the question is if you have tried opening first in France only, so that you don't have the CTR review,
right? A single country.
and then
I don't think I would do that. I would still use the CTR because any changes you're going to make, you're going to have to keep going back to that. And so I would try to pick a country outside of CTR and outside of the US. I would try to use the CTR. Yeah. And I would try to use the US. It's changed a little bit the patterns for that, but so that way you have the flexibility. If I know I'm going to slow down here on my 3 main CTR countries, I can still do a few things over here. We're changing, like for example, we're changing our dosing schedule a little bit, and we're trying to quickly do it here and then wait for it there and then just get everything to come into balance.
So we talked about the preclinical data. We talked about the submission and the strategy on which country and how to make it what consideration to do to try to accelerate. How do we get the fastest possible to some clinical data that can be interesting for, and we're talking about phase 1. And so what do we mean? We mean that either we have strong data that will develop the drug ourselves, or that we have strong enough data that the large pharma wants to partner, and that we have good enough data that investors will keep funding our project. I want to make a parenthesis here. I was a panelist on the FDA ASCO Project Optimus 2nd, and we had lots of pre-meeting alignment calls. And then I've attended a couple of public panels run also by the FDA. When we talk about these things among developers, we tend to go to the practical points and say, okay, how do we do it faster? How do we make it cost efficient and so on? And I realized that when you hear these discussions from an academic point of view, then they say the dark word, the dark pharma. Oh, you think about the money. Let's be clear. We think about the cost because we actually have to pay for these things and we have to make these things happen. We think about being fast because the longer it takes, the more it costs. But also, in my view, I've been a practicing oncology. I was an investigator. Phase 1 oncology patients are different than other patients. They have 3 months to live on average. And they're probably giving us their last or second from last chance of trying something. The investigator is convincing them, okay, we're underdosing this drug because it's dose escalation. We don't know if it's going to work, but this is probably the best chance you have at this point in your life. So do you want to gamble the last 3 months on this? And so at least most of us, hopefully, are very conscious that there's a patient on the other side. We have to make it as fast as possible because, one, we don't want to drag a study and ask more patients to come on something that maybe is not a drug. 1 out of 10 experimental molecules makes it to the market. And 2, we need to be fast because those patients are dying, and so we need to give drugs. But yes, there's also economic considerations because biotechs are running out of money. And if 1 out of 10 may be a drug and 10 companies go under, we're going to lose that one drug. Yeah. It's never going to be developed. Yeah. So when we talk about money and costs and speed, it's, of course, the patient is first and foremost. But when we talk practically, we may not realize that an academic listening to this says, you're the dark pharma. No.
Yeah, the practicality. Yes.
With developers.
The practicality. So everything for me is seamless and as efficient as possible. Okay, so and for Project Optimus, we're doing backfilling rather than prospective randomizations because that's slower, and I'm not going to slow down for that. So I'm backfilling, but I'm replicating what a randomization would be. You don't have to have the randomization. You just have to need the multiple levels and the different types of levels. So that's one example. Second would be we're using a micro-cohort approach. Okay, and I totally agree with you. Strictly define your patients. So, for example, I'll give you the example. We
have 5 minutes, so we need to be efficient also in how we run this.
I'll give you a quick example.
Or they cut the lights.
I'll give you a quick example. We're in HCC right now. We had some public data on that, and we're strictly defining the patients we're bringing in, very strictly. Were they prior exposed to IO or not? Under what conditions?
So how long? Populations.
And we're very strictly defining that. So what we do is we'll say, I have a few micro cohorts, and I will very strictly define them. In my protocol, I say I can stop any cohort at any time, which gives me flexibility.
So you have a continuous assessment of efficacy.
Like an adaptable design, which is an adaptable design. And then I put my futility analysis there in a very smart way. I put it low so I don't have to stop too soon if I don't want to, but I can also have a higher threshold if I want to stop and be very reasonable about that. I put flexible language there so that if I want to kind of alter and I say I want to start restricting only to the folate receptor patients now or not or something, so I have some flexibility so I don't have to pause. And then we just look at a series of micro cohorts and we just stop
I
the
love
ones.
the micro cohort concept. Actually, one of our companies, Pharmaset, they were acquired for $11 billion after, a dozen different micro cohorts of 10 patients at a time. So again, if you can clearly define the patients, right, and see, again, high thresholds, I think, as well. In that situation, it was cure of HCV, which was a very clear endpoint. I think that's an incredibly valuable tool. And again, it was enough to get Pharmaset acquired for $11 billion.
And Havitz is back, so he's going to whip at us if we don't conclude the panel. But I think, so we have it. We have what the discussion was, how do we accelerate things? How do we get quick to data in today's world? And we discussed the preclinical side of this.
One other thing I was going to bring up, maybe I would love your feedback on it too, is I'm seeing a lot of companies keep their clinical sites open and kind of roll in from 1B to 2A. And I think that a major part of the overhead, it appears to me, is really the opening of sites, the training of investigators. So again, a lot of people are kind of doing cohorts, or let's say a mini 1B, and then rolling straight into a 2A. Those sites are ready to go, and those are essentially the expansion process jumps right in.
That's an easier transition than a phase 1A to a phase 1B. Because in a phase 1A, you have all comers patients and you have pure phase 1 site. When you decide, I need to go into lung and breast cancer, the phase 1 investigator...
I agree, that's different. From 1B to 2A or 1B to fit 1B expansion. So that's something we're seeing a lot. And that really can save you like 6 months
for
of
me,
opening and closing sites.
For me, it's all seamless. Okay, so we'll go from the 1A to an expansion, as long as we follow the expansion cohort rules where you have to have a stopping rule. Otherwise, you get an independent monitoring. We don't want that. That's going to slow things down.
Plan it in advance.
Yeah, you have to say what your stopping rule is going to be. And then the FDA will let you go to an expansion, okay, if you clear the futility. And then you go back to the FDA with some kind of pre-breakthrough or breakthrough type of discussion. And then they'll say, go ahead and amend and allow yourself to go even further. So for me, I'm always going from 1A to 1B to 2A to 2B. It's all seamless. And I don't want any pausing in there. And I like that. We keep leveraging the sites. And we talk to sites to see if they're interested in those types of cohorts that we're expanding to later.
A couple of caveats are that the European new regulations will make it much harder to add indications unless you had planned, and combinations unless you had planned in the protocol. And also, you mentioned the absence of IDM, an independent monitoring committee. In US, yes, but many European countries actually do not allow you a protocol with phase 1B expansions unless you have.
Well, I saw that at Servier and I was surprised, but what it really was, which I found, is if you put your stopping rule there, then they won't ask you. Spain in particular always asks, for sure.
France.
But if we put our stopping rules there, and they were like non-binding stopping rules, I said, well, that's not a stopping rule. Stopping rule is a stopping rule. So you say, if I don't see 1 in 10 with a response, then it stops. And then that's how we got around it.
All right. Thank you. Thank you for having us and not kicking us out. Good to go.